new technologies, and this is the first time NODCC's had to try to figure out how to stream. So there may be some glitches along the way, and we are the trial run. So thank you for your patience today. I'm Lynn Paul. Many of you know me. I've been around for a while. And I wanted to start today by asking you guys a few questions. First, how many of you went to one of the family gatherings between 1999 and 2002? Anybody? Kristen? Warren, raise your hand. How about anybody who's been on the NODCC board or the OSTOC board or Corpal? How many of you have come to conferences in between 2002 and 2010? Right, early on. Oh, you're hard to hear? Sorry. This is what I need. I said, how many of you have been to one of the conferences from 2002 to 2010? Okay. So a lot of you are fairly new. How many of you have had days where you thought, my life would be so much better if we didn't have to deal with this whole ACC, DCC thing? How many of you thought it's just like we're fighting all of the time? Fighting to get somebody to understand what this diagnosis is, fighting to get teachers to pay attention, fighting to get resources, fighting to even know what's going on, fighting my own fear, fighting all of the frustrations of time and energy. Anybody? Come on, feedback. I want to see. Honestly, yeah. Part of what I want you to know, and the reason I started this way, is those feelings and those frustrations and that drive to figure this out, to overcome, to keep going, and the days are really hard and long, is something that it's easy to think you're alone in, right? And so you come to these conferences so you can be with other families and you can be with other people who get it. And it may come as a bit of a surprise because we as scientists are not always emotionally very sensitive and present, even though I think we're among the better of those. But you'd be surprised how much this group of scientists gets it. And I'm saying that not just because I was at the first meeting in 1999 in a sleeping bag, in a tent, or in a cabin at a Easter SEALs camp. And I paid my way and got there and spoke. And then Warren came and we sat around late at night in 2001, figuring out how do we make an organization. We were inspired by the ACC network, the families that bonded together and said, this is a problem. Somebody's got to help us. And since then, I think it was 2003, so almost 20 years ago, that I went up to meet Elliot because he wanted to come to one of these conferences. And so Dr. Scheer wanted to be a part of it. And I said, I want to know who you are because I don't want to introduce you to our families unless I trust you. And he thought I was crazy. And I could tell he cared. And that makes a difference. And through the years, you've heard the three of us talk ad nauseum, probably, different sessions over time. And then starting 10 years ago, there was OSTOC starting to develop, which was very exciting. And I remember another really important conversation in 2013 with Dr. Richards. First time I met her, and she said, they want to start a family organization in Australia. I study mice. Well, I don't know how to help families with, and I was like, but you care, you can do this. And she's knocked it out of the park. She's been beside them the whole way, and they have a champion there. And she does study people now. and has expanded and followed the questions. And I would say that's what a good scientist does. We follow the questions and we follow the things that matter. And so every time we come to this meeting, we learn more about what matters. And we get inspired to keep going because there's not a lot of reinforcement and a lot of reward in this career, to be honest. People don't go into science to get rich or to have fame. You do it because you don't mind staying up all night and writing grants, and anybody who knows you thinks you're crazy for doing it. So you got to be kind of compelled, just like all of you. You stay up late at night, you keep going, you keep badgering your family and friends to understand this, you keep badgering doctors. This is a race of endurance. And I just really wanted you to know today, you're not alone in it. It may be frustrating that there aren't more professionals who get it. There aren't more doctors that know the diagnosis. There's not literature sitting around in every waiting room. But we're with you. We're small, but it's a pretty mighty group of scientists. You've got, well, Linda was the president of the Australasian Society for Neuroscience. She's the head of neuroscience at Washington University. You've got people speaking today from Germany and Australia and France who are leaders in their institutions, who most of us, if you looked at the portion of our time that is officially for doing this work, it's anywhere from 1 to maybe 10% of our time. If that, if you looked at what our jobs think we're doing, but we all carve out time for this because we care. And it's kind of frustrating sometimes, like you guys, we wish we could do more. And one of the coolest things about brain science right now is it's moving so fast. The rate of discovery, the rate of new technology, I mean, it's amazing. And all this communication, the ability to meet. Yesterday, we had 4 hours of meetings and we had people from 12, 15 countries. Fernanda, I forgot about Fernanda. Fernanda helped set up the family organization in Brazil. Dr. Booth is on the board of the group in the UK. Tanya, who also does fetal pathology, a few years ago, ended up being the scientific director for the group in France and championing that group. And we each love our groups. We each care about them and want to do everything we can to help you and to help you guys connect with each other and all of those things. So there's so much that could be done. And in 2015, we had one of our big dreams come true. We met in Australia. Linda was able to arrange for a bunch of scientists to come together, and we sat down and said, what do we want to happen? What are your questions? What are your dreams? And we all tend to be visionaries and come up with big ideas. If we were entrepreneurs, it'd be a different world. But we laid out a plan for working together, for researchers to share. I have 3 families who have this condition, and you have 2 families in Australia that have this condition, and there's 1 in France. Well, that suddenly gives us 6. Well, 6 is enough that we can start to make some inferences. We can start to understand this diagnosis, this genetic condition. You see what a difference it makes to go from 2 to 6 families that have the same genetics? It's small, but it makes a difference, and it gets something published, and it helps those 6 families. And a lot of what we're doing is trying to get all of the information we can together about all of the people around the world that have this diagnosis. We pull it all together, then we can start to use all those fancy technological things and statistics and artificial intelligence to make sense of why are some kids really impaired and some kids aren't? How can we figure out what predicts how somebody's going to do? You know, that's the holy grail here. Like every family wants to know. Everybody who's diagnosed before birth wants to know what's going to happen. There are really fancy, amazing ways to start to answer that question. And it comes with gathering all the information we possibly can about all the people who have this diagnosis and following them on as long as you'll let us keep asking you questions and putting all of it together so we can work together. And the rate of discovery from that is going to be like shooting the moon. Can do that so much more quickly. So I want you to have hope. If I look over the grants that have come in from our main resource for funding in the United States to fund research is from the National Institutes of Health. And I was going to show you some really boring slides about how many years it takes and how many times you have to apply and the funding rates. And, you know, if you only get 12 to 15% of the grants get funded, and then you have to resubmit, and it takes a year to resubmit. And then my colleagues kindly reminded me that we're terribly discouraged. Why would we share that with you? So I'm going to not give you all those details, but I will tell you that when I pulled up agenesis of the Corpus Callosum grants or dysgenesis, the number of them was increasing. And we got our first grant to study behavior in this condition in 2018. So part of me, you know, is frustrated that it's the first time, but on the other hand, hey, it means Jasmine's got a job and can do all those Vineland calls and talk to you, and we get more information, which is awesome. So one thing I wanted to say to you this morning is you've got a great, really great team of scientists out there around the world behind you. There is movement, the number of publications, people are starting to recognize this diagnosis. So there's progress. There can be a lot more progress if we have the tools we need to do it. And if all of you as family members and as people with ACC bond together and say, I'll give my hour to answering questionnaires. I'll sign up for the registry and put my family information in there so that my kid's diagnosis counts. Because you know how it is. You say there are 10 people with this condition and everybody goes, eh. You say there are 10,000 and they go, well, maybe we should study that. Maybe we should give some money for that. It's kind of logic, right? So I'm preaching to the choir, as they would say, talking to all of you in this room. But I'm also hoping that you're apostles, that you will go out and spread the word, that you will tell other families this weekend, sign up for the NODCC registry. Doesn't matter what country you're in, we're all in this family together. Sign up for Dr. Scher's study, sign up for Dr. Richard's study, sign up for the Caltech and Fuller studies. Give whatever information you can. And if you don't qualify for that study, tell other people to sign up for it. And we'll just keep trying to do what we can do. And if you know people who have scads of money, our science group would love to have some resources so we can meet more regularly and we can share data and all of those things. In the meantime, we will use duct tape and chicken wire and keep doing what we can with the tools we have. So those late nights, when you're sitting up and you're thinking, dear Lord, why me? And you think, oh, there are other mothers out there, there are other parents out there. I just want you to know that it may be that Elliot and I are on Zoom, both of us falling asleep, working on a grant at the same time, because it's happened many times. At least you're not in your office alone because somebody else is in their office. There are people out there who care, and we'll keep trying to get you answers. So today, as you listen, I hope you appreciate and enjoy all of our speakers, and learn things, ask good questions, ask whatever you want. We're here for you today. Keep giving us your ideas, and we thank you for letting us be a part of your lives. This has, like many of you, been a vacation for our family, my husband who does the AV, and it's our community, and that's part of why we keep coming. Not because it's our job, because for most of us it isn't. It's our side gig. So we're here, and I thank you for letting us be a part of it. So this morning, I'm going to introduce to you Dr. Linda Richards, who is, as I mentioned, the scientific advisor for OSDOC. She was the associate director for the Queensland Brain Institute in Australia at the University of Brisbane, and is now the head director of neuroscience at Washington University as of the last year, and has come to love St. Louis. She's lived in the United States before, and as an Australian, I think she tolerates this fairly well. And she has going to give you kind of the big picture of what we're looking at and why what we do might impact you and why we think it's important for the world to know. So have a great day, enjoy the conference, and I'll talk to you all again later about what we're doing with that grant that I mentioned.
Good morning, everybody, and good morning, everybody who's on Zoom. Thank you so much for having us here. I'm Linda Richards, as Lynn introduced, and I'm here with my team of scientists as well. And we have some Fernanda Tovar-Moll, who's also here as part of the board of the IRC5 with her team. And we are really excited to talk to you and meet you and learn your stories. So I thought what I might do is try and give you a bit of an overview of why we do science and why does it matter. So do I advance the slides with, there we go. All right, so I thought, let me just start with a couple of definitions because these are maybe helpful as I'm speaking. We do try and we speak different languages. I mean, I speak Australian, for goodness sake, but also we do speak different languages sometimes. So DCC, disorder of the corpus callosum, sometimes we call it corpus callosum dysgenesis or a corpus callosum disorder. And I know that the acronym is very important. So let's just say DCC equals CCD. And we can talk about, you know, what are the nuances of that. But if I say CCD, I mean DCC. I also think it's useful to know, what do we mean by cognition? If we're studying cognition, what is that? And you can think of that broadly in terms of mind. So we have brain and mind and how we think about things. And then another word you're going to hear a lot is phenotype. And it will come out of my mouth because I can't stop it. But a phenotype is basically an outcome of a change that's different from neurotypical. You probably heard the term neurotypical. And a phenotype is the outcome of a change in a gene, a change in a structure, or a change in cognition. And like a change in a structure, that's where you get into the details of, is it agenesis, complete agenesis, partial agenesis, hypergenesis. These are phenotypes. Okay, so that's some definitions, and just bear those in mind. I'm not sure how, do you just press the space? How do you advance the slides? We've had a few technical difficulties, sorry about that, but maybe, sorry? Yeah, the arrow buttons are not advancing the slide. Yeah, maybe escape out of it and then go to the third slide. I wonder if it's because it doesn't have. Yeah, I think it's because it doesn't have PowerPoint on this machine. So these were going to come up in sequence, but I'll just run you through them. So I thought it's also good to give a bit of a definition about scientists and doctors. What do they do and why are they important and different? Scientists are just obsessed with trying to figure out how things work. Our major goal, what drives us, what is our passion is discovery. We want to discover why, what, and how. Or what, why, and how. Discovery leads to progress, right? And progress is really what I mean by that is understanding what, why, and how. And we go about this by measuring things, quantifying things, and providing evidence. And that's what we grasp. And so understanding is evidence, right? So we gather more and more evidence. And at one point, we gather so much evidence that basically it's a fact. And that evidence is what leads to new treatments and an understanding of what caused the disorder and how. And effective treatments require evidence base, a base of evidence that that treatment is going to work. And why is that treatment going to work? Because we understand what went wrong. And that is what we're tackling. So that's a scientist. You cannot make progress unless you do science. You can't make progress in understanding things unless you do science. Doctors and therapists have a different role. And by therapists, I mean like allied health professionals, speech pathologists, physiotherapists, occupational therapists, all the types of therapists that everybody here is familiar with. They implement the therapies that come through research, scientific research, which provides the evidence for those therapies, but they are the people that provide those therapies. Now, there are some very special people in the world that manage to be both a scientist and a doctor. And most of the people that are involved in the IRC5 happen to be scientists and doctors. I'm not a doctor. I'm just a scientist. But there are some advantages in only being a scientist because I can spend all my time figuring out what's gone wrong, how, and why. Whereas they're juggling implementing the therapies and trying to come up with new things. But also, why would they do that? Why would somebody, spend all their time doing two different jobs? Because they recognize that in order to help their patients, they have very little to work with. So they want to be part of the solution and identify what are the new therapies. So all this comes down to a simple fact, and that is that if you don't understand what is wrong, you can't treat it with evidence-based effective treatments. And that only comes from research. I think we're going to have to do the same thing again, Scott, sorry. So the next thing I thought might be useful, and this is, please ask questions as well. If you want a clarification or just interrupt me. Yeah, I think it's because I had some PowerPoint thingies in there. So the current challenge in disorders of the corpus callosum, there are many challenges for you. And this is a way that I think about it. Part of the challenge is because disorders of the corpus callosum are vastly variable in their range of structural, genetic, cognitive phenotypes. So that makes it complicated, right? Because not everybody's the same. It's really quite vastly different. So if you're thinking about another field of medicine, you know, where you're trying to understand even breast cancer, let's say breast cancer, really, you know, important medical problem, it's much more defined. So here we're tackling something that is vastly more different and difficult. So because there's this huge variability, it makes the challenge of understanding and treating disorders of the corpus callosum very difficult for individuals with DCC and their families and doctors and scientists. So that's the current challenge. And oh, yeah, good. All right, so let's go back one. Right. And so what's the challenge for doctors? And I'm not talking about the doctors that are here. These are the doctors that are experts in disorders of the corpus callosum. I'm talking about the doctor that you will go and see who knows nothing about disorders of the corpus callosum, which is the majority of clinicians that I've come across. So they can provide a structural diagnosis, but they don't really know what that will mean for you or your child. And usually doctors don't know exactly how to treat your specific DCC phenotype, be that genetic, cognitive phenotype, or structural. And usually, of course, they have to resort to therapies that work for other conditions because we don't have currently very, very specialized, maybe there are some, but they have come from the people in this room. But those specialist therapies are what will come from the research that Lynn was just talking about. And often they're unable to provide much guidance or prognosis, like what will it mean for my child? That's a prognosis, right? And often that's not possible because currently that kind of information is not yet available. And the reason is because we haven't done longitudinal studies like the one that Lynn is currently conducting over time so that we can get that information and provide that as a prognosis. And the reason that the IRC5 is so important is because it's an interdisciplinary group of people that have come together that are specialists in genetics, imaging, brain imaging. I'm going to talk about brain imaging in a minute. It's just awesome. And then cognition. So Lynn is a cognitive neuroscientist. Warren is a cognitive neuroscientist. So people who specialize in cognition. And then there are also people who specialize in brain development. That's my specialty. How does the brain develop? And then, you know, people who also work on animal models. And I'll tell you why animal models are so crucial here in a moment. So the scientific challenge for us walking into this is, what does the corpus callosum actually do? That is a question still very much unanswered. The answers have come from the people in this room and from very important but older work that was done prior to Lynn and Warren, and people that studied the corpus callosum from a very different angle, people that studied, scientists that studied people that had their corpus callosum cut because they had intractable epilepsy, and then some scientists studied those people. They are known as split brain patients, and they have a completely different phenotype to people with a congenital abnormality of the corpus callosum, a disorder of the corpus callosum. So that work, although it's important and told us something, it doesn't have as much impact on understanding the cognitive outcome of a disorder of the corpus callosum. So how does the corpus callosum underpin brain function and cognition? That is a really important question. And it's a question we are trying to address as a group. And that is what's going to help us pinpoint what kind of cognitive therapies are going to be most effective. And then how does this change throughout life? Again, going back to what we call longitudinal studies across the lifespan. How does a disorder of the corpus callosum affect you as an infant, as a child, as an adolescent, as an adult, as an aging adult? How does it affect you at these different life stages? Something I'm very interested in is, how do disorders of the corpus callosum arise? What happened? Was it environmental? Was it genetic? And if it was genetic, what did the gene do? How did it affect the development of the brain? And what genes are involved? So we have specialists in genetics and within the IRC5 and within the community, who are identifying genes that are related to different disorders of the corpus callosum. But identifying the gene is only the very first step. It's not much use if we don't know what the gene actually does. And that's where animal models actually come in, because the only way we can understand what a gene actually does in brain development is to disrupt it in an animal like a mouse. And a mouse actually has a corpus callosum is very similar to a human. And we can disrupt it in the mouse, and then we can study exactly how disrupting that gene in a mouse affects the corpus callosum. And then we can look at ways of modifying that to see if we can change the phenotype. We're also interested in what other conditions are associated with a disorder of the corpus callosum and why. So why is autism associated with, some people have autism, some people don't, who have a disorder of the corpus callosum? Why do some people have epilepsy? Why do some people have movement disorders like cerebral palsy? Why do some people have changes in their brain structure that aren't the corpus callosum, but there are other changes? How do those come about? Are they related to the corpus callosum, or are they completely separate? Because if we want to treat that, then we need to understand exactly what's the cause. So these questions, these are questions that are important to this group, but honestly, they actually lie at the heart of scientists striving to understand fundamentally how the brain develops and how it functions. So this work has even broader impacts in generally understanding the brain for humanity. And it's quite, whoops, let me see if I can. All right, so let me just move into a little bit about why I'm so excited about the brain. So the brain is just an amazing organ. It is unique in every one of us, because every one of us has our own lived experience, and those memories are contained within your own brain. And we are excited to understand how that happens. And can we really understand how the brain works? Oops. So your amazing brain normally has, yep, we've lost it. But what that picture, it's not normally just a gray brain. It has all these different areas in the brain that are important for reading, mathematics, creativity, motor skills, thinking, planning ahead, language, all of these different functions, emotions, your ability to love somebody, hate this talk, whatever it is, and all your sensory information, every piece of information that's coming in through your eyes, your ears, your nose, your mouth, all of that is processed in your brain. Now, how is it all put together? The corpus callosum is the largest fiber tract in the brain, connecting the two hemispheres. It is enormous, and it is very important for processing all of this kind of information. So it's not simple. It doesn't do one simple thing. We know that people with a disorder of the corpus callosum are actually very special, because what happens during development, and this is the bit I know something about, what happens during early development when the corpus callosum fails to form in the neurotypical way is that the brain actually reorganizes itself. Now, we have evidence of this. And we have evidence from brain imaging, and myself and one of our colleagues from Vienna will be talking about this in the next talk. But we now have really super sophisticated ways of looking at the structure of the brain, far more sophisticated than the average MRI you would see from a doctor. And these are actually only available in research settings. So we use high field magnetic resonance to image the brain in very great detail, and we can look at the connections between all areas of the brain. And I'm going to see if I can move on to that. Now, unfortunately, this is not going to run. I'm not sure how much you can see on there, but afterwards, please come up and I'll show you a beautiful movie which steps you through these connections throughout an adult living human brain. So what we can do is use this special type of MRI to look at which areas of the brain are connected. And we can color code the connections based on whether they go front to back, side to side, up and down. And that's what all those colors are in that image. The corpus callosum, the ones going left to right or right to left, are red. And so if I could step through that movie, you would see all of these connections throughout the brain. Now, this is a neurotypical brain. We can also do this in a person with a disorder of the corpus callosum. And our goal, our hope, is to ask this question: does the structure actually underpin the function? Or can you get the same function from all sorts of different structural rearrangements? Our current evidence, which is pretty scant right now, but our current evidence is that the brain is actually able to function pretty well despite having a completely rewired structure. Now, what we've also discovered is that it doesn't rewire the same in every person that has a corpus callosum disorder. So using this kind of technology, we can actually see those connections. And what we'd love to do over time is to pair that with the cognitive outcome. And then we'd also like to pair, so the structure with the genes. So if you have a genetic mutation Does it affect which part of the connectivity? What's disrupted and how? And then how does that affect the cognitive outcome? That's kind of IRC5 broadly speaking. Now, in order to understand the genes and how they affect the structure, we need animal models. So mice have a corpus callosum. Oh, here's the corpus callosum. Everybody's seen one. Yep. So we are all of us interested in how do disorders of the corpus callosum occur and how do they affect people. Now, I've just moved from Australia. My team here, they're all Australians. This is half the team. Have also all just moved from Australia, where we used to work with the Australian Disorders of the Corpus Callosum group in person. We are still working with them remotely, and we are super excited and hoping that we can work with you here in the US. We're now located in St. Louis at Washington University Medical School. Why did we move? Because we found an amazing place in WashU to do our research. It's an incredible medical school with a lot of resources. Now, that's not to say that everywhere else in the US that's doing this research or anywhere else in the world is also fantastic. What we hope to do is to get more researchers interested in trying to understand disorders of the corpus callosum. Because the more researchers we have, the faster the rate of discovery will be. All right, so, oops. So let me see. Yeah, I'm not sure this presentation is going to work, but so maybe it's useful to know a little bit about how science is done, and Lynn alluded to this. So let me just give you a quick, brief view of what's been happening with our group. So in moving here to the US, actually we had to give back all of our Australian funding for our research because the Australian government wouldn't allow us to move it. So here we are now applying to the National Institutes of Health, which is the major funding body in the US for health-related research. So in October, we plan to submit a grant application to the NIH in the hope that we will be one of the about 10% of applications. That's 10 out of 100, every 100 applications that have submitted, we are hoping that we're one of those 10 that get funded through that program. Firstly, of course, we need to build the group. We then need preliminary data. We need to have a really great idea, and then we put the grant application together and we submit it. It's then reviewed by other scientists who look very carefully over all of the scientific aspects of the program. And then if we're lucky, we get one of the grants. The grants usually last for up to 5 years, and we start our work. Now that will start in terms of building a group of people, trying to identify who would like to be involved in our research, asking you to consent for us to contact you, and then enrolling you in some of our studies. So we already have our ethics approval here. And so of course we needed to apply to a larger body that provides approval. They look over very carefully all the experiments that we want to do, and they look at it from an ethics point of view. Is this an ethical scientific program? So we have obtained that permission to do these experiments. And in fact, the team bought a few of our cognitive tasks with us. If anybody would like to come and have a look, we can show you exactly what we do. And we can explain to you, if you do this task, this is the kind of information we're trying to get out of it. We're trying to understand questions like, how are you thinking when you make a decision to do something in the game? And we're trying to understand how people with a disorder of the corpus callosum might be making decisions, doing decision making that might be different. So these are just some of the things that we're working on. Another really important question that Lynn brought up is, how can we speed up the pace of discovery? The way we speed up the pace of discovery is working together with you, and trying to get more people interested in working on this group of disorders. That's really how we're going to speed up the pace of discovery. And trying to get NIH interested in funding this kind of area of research. So we do a lot of advocacy, not just to try and draw attention to this group of disorders as being so crucially important for understanding the brain in general. Yeah, okay, that's not coming out. So what we'll be talking about in the next session is some of the more structural changes that occur in a disorder of the corpus callosum. So I won't go through that now. But one of the things, just as an example, that our lab has done over the last 20 years, I started working on the corpus callosum in 1997, was to identify the cause of complete agenesis of the corpus callosum. So we know how it occurs, and I can explain it to anybody that would like to come and talk to us later. What I was going to show you there was a mouse. All right, this is a mouse brain. I don't know if you can see it. Maybe come and have a look at the slides a little bit later, but mice also have a corpus callosum, and we can see probes bundles. You might have heard of these things. This is where the axons come. Okay, so a neuron has a long wire. It's called an axon. The axons come down and they can't cross. So they grow instead either front to back. And mice, this happens in mice too. Now, what's great for us is that then we can study this in a mouse. We can't do these kinds of experiments in people, but we can study them in mice. And we now have very, very sophisticated genetic ways of manipulating the mouse genome, so that not only can we take the genes that Elliot and others in our consortium identify, we can make the exact same mutation that occurs in people in a mouse, and we can see what is the effect of that. What does it do? How does it function? How did it cause the corpus callosum disorder? All right. I think that I might wrap it up there and ask if you have any questions that you would like to, anything that didn't make sense, or any questions. Yes, please. Yes. Yeah. Absolutely. Yeah, so I'll just repeat your question for the people on Zoom because they can only hear what comes out of the microphone. So the question was, do we do research on babies and adults all the way through the different life stages? And the answer is, we actually start before birth. We have specialists in the International Consortium in fetal development, because the corpus callosum actually begins to form between 12 and 20 weeks of gestation. So the disorders actually start to arise around 12 weeks gestation. So we have prenatal experts as well. And then we have people working on babies, infants, all the way through adolescence and into adulthood, and older adults as well. That's a good question. Yeah. Just one sec, down the back there. Yeah, so the whole, one thing that I'm really excited about is that NODCC is setting up a registry, and that registry is where you can put your information. And my understanding, and I see Lynn Snyder here who told me about the registry, my understanding is that rather than you signing up with my study and Lynn's study and Fernanda's study and everybody else who's on Zoom study, you sign up once to this registry, and then we will be able to access that registry. And what we would do then is to look, so, you know, it depends on which kind of study that we might be able to do, but then we'll be able to find people that have fit that criteria for the study. And then because you will have consented that we can contact you, then we can contact you. Yep. And what's great now is that you could go to San Francisco, you could go to St. Louis, at least. They're the two places, and you could go to Boston. I'm not sure where you live, but we would be able to try and find a place where you could get one of those scans. Okay, one here, and then one here. Totally. Yep. Yeah. I'm actually going to give a talk on Saturday. Repeat the question. Sorry. Thank you. The question was, how many different kinds of mutations are we working on in this scientific consortium? So one of our colleagues in France, Tanya Ati-Batash, who'll be on in a few hours in this session today, she actually made a callosome, we call it. It's got 400 genes on it already. So we're talking about vast numbers of genes that are involved in formation of a corpus callosum. And at the moment, so Elliot is also working on, we have a whole genetics group working on all of these different genes and just identifying them. What are the mutations? Are they actually causing the disorder? So yeah, we're working on lots of genes. A quick follow-up, yeah. Yep. Yeah. So it's a good point, because there are different subgroups. As I said, the disorder of the corpus callosum is a vastly heterogeneous, different. There are lots of differences, and there are a lot of different subgroups. And as the genes get identified, then it is a way of kind of grouping them. And then you get subgroups that are splintering off that have different, specific gene mutations. Our job is to try and figure out how do they all work together, because the genes all don't work in isolation. There are pathways. There are multiple genes in a pathway. So don't think about them as one gene, one phenotype. Lynn.
I promise I didn't ask her to ask that question. The question was, how do you as families advocate for NIH funding? Write to your congresspeople, write to NIH. The branches that we mainly get funded by are the National Institute of Neurological Disease and Stroke. It's called NINDS. Elliot's got grants with them. and the National Institute of Child Health and Development, and ICHD is where I have mine. So those are the two of the main branches that would fund our work. So you can contact, they have directors, and you can say, this is an important topic, blah, 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 blah. They're human beings, right? So the more they hear a topic come up, the more sensitized they are to hear it. And the more likely they are to pay attention when that grant comes across their desk. NIH gets a budget from Congress. Congress has to approve their decisions on all of the grants that they give.
This is why there's so much funding in autism and why we're now really making progress in autism. Because the families got together and said, we need research on this topic. We need to understand what's going on and how we can help our children. So that's the kind of advocacy that does make a difference. There is no doubt that makes a difference. Question down the back. Yeah. Yeah. So a couple of things in that. The question was, what about chorionic versus blah. Yeah, Elliot could come up here and talk about it. CVS. So the issue there is we're mixing up structural diagnosis of a corpus callosum and genetic diagnosis of a corpus callosum disorder. So you can't see a corpus callosum by ultrasound until at least 18 to 20 weeks gestation. You just can't see it. And by definition, that is the definition of a disorder of the corpus callosum. You got to be able to see it. However, we do know some of the genes that cause disorders of the corpus callosum, and that is what you would screen in a CVS sample. If you knew what they were and you had actually another advocacy thing, right? Because in order to get that test paid for by your health insurance, you would need to, it would need to be a robust test that they would do routinely, right? Do you want to say anything about that, Elliot? So you're going to hear today, you'll hear from the genetics team, the imaging team, the cognitive team. So it'd be good to ask those specific questions then. And I think we have to wrap it up.
We're going to need to wrap up soon, but we have a couple of questions from Zoom, and this one tags on to that. How does one partition DCC outcomes related to gene deletion versus mutation?
Yep. Yeah, how do you parcellate gene deletion versus gene mutation? So that is, again, that's like a genetic definition. And I think that would be best discussed when we talk about genetics in the session on genetics within this day today. Yeah. So that I'm not answering the question, sorry, person on Zoom.
I'm going to give the other question from remote. Are there data that suggest that DCC can be mitigated by neuroplasticity, and to what extent? And I'll take a first stab at that. As Linda mentioned, we know that the brain can reorganize, and it does. If it can't grow one way, it'll figure out another way to grow. And then each brain does it a little differently. So the fact that you can function as well as you can without this huge structure says, yeah, plasticity can make a difference, but it's varied. And a lot of times it's using structures that would have been there, but would have been like the backup system. So the brain says, oh, there's a backup system. Since it can't do that, we'll recruit that and use it. And that is one of our big questions, is being able to figure out and identify what backup system's being used, how has it been recruited, how effective is it, et cetera. All right. We'll take these last two questions and then we'll wrap for this session, have a little break and come back.
Yeah.
I'll jump in there and I'll talk at the end of the day about the longitudinal study that we've started working with infants and through childhood. Part of that study, most of it's done remotely, but about half of the people who participate are actually going to be tested in person. And this is part of this research process. While you're in Minnesota being tested, we are trying to get a small grant within the institution to pay for the MRIs so we can get pilot data so that we can put in a bigger grant to do MRI. And there's huge studies and at Minnesota looking at autism this way. So we have access to all these comparison groups and these paradigms of doing the research. We're just going to have to get the pilot data so that we can get funding to actually do the same thing in ACC. So that's spot on what we want to do.
These scans are very expensive, I should say. So we pay for them from our research grants if we have research grants. But maybe you know how much the scans cost, Ryan, at WashU, but in Australia we paid $700 a scan. That's just to give you a ballpark of what the kind of grant funding is required. So if you have, 10 participants, there you go, $7,000. So it's, it's expensive to do this kind of research. To sequence a human genome costs about $1,500 now, but that's just to get the sequence. To pay somebody to actually look at all those letters and figure out where the mutation is, then you need a salary of somebody called a bioinformatics person. So it's, it's vastly expensive to do this kind of research, just FYI. Yeah. And there was one more question and then we'll break. I think you had the question and then we'll break, and then please come and talk to us in the break. And then we've got more cool stuff to show you. Yeah, that's what we're working on. That is what we really want to understand even more so. So the question was, there's a structural diagnosis. There could be a genetic diagnosis. Is there a cognitive diagnosis? So there is a cognitive diagnosis, and this CCD syndrome was actually discovered and put forward by Warren Brown and Lynn. There is, these are like the core syndromic kind of aspects of the cognitive phenotype. But there are many, many, many aspects to this, and that's what the whole consortium is also working on. So can you specifically phenotype somebody with a corpus callosum disorder and be able to pick them out? They probably could. But your average, clinical psychologist, no way. Yep. Okay, thank you.